This lesson is on the levels of structure of proteins. We're going to talk about primary, secondary, tertiary, and quaternary structure. First, primary structure. Primary structure of a polypeptide or of a protein is just the sequence of amino acids. So remember that polypeptides, which make up proteins, are just polymers of multiple amino acids joined together by peptide bonds. So the primary structure is just which amino acids are there and in what order. The secondary structure involves repeated patterns called alpha helices, singular being a helix, which is a coil-like structure, or beta pleated sheets, which is a kind of folded sheet-like structure, and these can repeat throughout proteins. So I'll try and draw a beta pleated sheet here like this. These structures are held together by hydrogen bonds between the parts of the backbone of the polypeptide chain. Again, they're held together by hydrogen bonds between, I'm going to write between like this, between atoms of the backbone of the polypeptide. So how does that work? Well, remember that in a peptide bond, which looks like this, formed from a dehydration reaction of the carboxyl group of one protein and the amine group of the other, you have a partially negative oxygen here, because oxygen is much more electronegative than carbon, so it steals electrons away. And you have a partially positive hydrogen here for the opposite reason, that nitrogen is more electronegative than hydrogen, so it steals some electrons away. If this hydrogen could come in contact with the oxygen of another amino acid or another peptide bond in another part of the protein, that could form a hydrogen bond. And so that would be the kinds of bonds that stabilize the alpha helices and the beta pleated sheets, these repeated motifs that form secondary structure. Let's talk about tertiary structure now. Tertiary structure refers to the overall three-dimensional structure of one polypeptide. So the overall 3D structure of a single polypeptide. So the polypeptide assumes a three-dimensional conformation, let's say like that, and that is a result of interactions that the side chains, or the R groups, have, either with each other or with the aqueous environment. And those can be of several different varieties. You can have hydrogen bonds between different polar R groups or side chains. You can have ionic bonds between charged side chains or R groups. You can have covalent bonds. For example, in the case of cysteine, with its sulfhydryl group, it can form a disulfide bridge like we saw in a previous lesson. You can also have what are called hydrophobic interactions. Hydrophobic interactions refer to when hydrophobic side chains try to get away from the surrounding aqueous environment by burying themselves in the interior of a protein. So you can have a variety of different kinds of interactions between the R groups or between the R groups and the aqueous environment that lead to this three-dimensional overall shape of a single polypeptide, and that's tertiary structure. Now, all proteins have primary, secondary, and tertiary structure. Not all of them have quaternary, or you'll also hear it pronounced quaternary structure. And that's because quaternary structure is the association of several different polypeptides into a single protein. So you can have proteins that are made of multiple polypeptides, and in those cases, that protein would have quaternary structure. In cases where you have only a single polypeptide in a protein, there's no quaternary structure. So an example would be hemoglobin. Hemoglobin, abbreviated like that, has two alpha subunits. Those are two of a particular kind of polypeptide, and two beta subunits. And together, all four of those subunits make up the single hemoglobin molecule. So you've got something that looks like this, with two of one kind of subunit and then two of another, and that makes a single protein. That's quaternary structure. Again, it's only in some proteins. Just one more thing to add here. You can see that the shape of a protein is determined by the different kinds of interactions or bonds that it has, and the shape is what is responsible for its function. Now, if you heat a protein to a high temperature, or if you change its chemical environment, then you can alter the way that these bonds happen, and therefore you can change its shape. And that loss of shape, and therefore the accompanying loss of function, is called denaturation. And so heating proteins or changing the pH, for example, can result in a loss of shape or function, and that's called denaturation. And depending on the situation, that may or may not be reversible, depending on whether the bonds that have been broken can be reformed or new bonds that have been formed can be broken. An example of denaturation that you'll be familiar with is cooking an egg and seeing the egg white turn white. That's actually denaturation of the albumin proteins in the egg. So that's an introduction to primary, secondary, tertiary, and quaternary structure of proteins.